Thanks, everybody, for coming. We have a great turnout today for our two exciting speakers. Before we get started, I want to remind everyone about the Mycotalk series at the end of the month, and I'll post the information on that in the chat. So please sign up. That's a great series of talks. And also, if you're interested in giving a talk here, then please submit an abstract, and I'll post the URL for it. The abstract submission into the chat as well. So our format is that first Matt Blango will speak, and then Carolina Coelho will speak, and we'll take questions at the end. So Matt Blango is a new PI at Leibniz HKE in Jena, Germany, and his undergraduate work was in biochemistry and molecular biology at Boston University, followed by a PhD in Matt Mulvey's lab at the University of Utah, where he worked on bacterial pathogenesis. He then did a postdoc in Brenda Bass's lab, where he learned about RNA biochemistry and bioinformatics, before doing a second postdoc in Axel Braga's lab at Leibniz HKE, where he learned fungal biology. And so Matt will talk today about his work on RNA therapeutics.
Cool. Thanks, Jessica. Let me share the screen. And I hope you all can hear me. Okay, so yeah, thank you to the organizing committee for giving me this opportunity to talk to you today. I'm really excited to share some of the work that we've been doing for the past few months, but really the past few years in the lab of Axel Bragaga while I was a postdoc and a small group leader. And I'll talk a little bit about the work that we were doing there, but also sort of give you the movie trailer version of what we're going to be doing in my own lab moving forward. And so my lab is now about 2.5 months old, so we're just getting started. And so what I would like to talk to you about today are two different topics. The first, RNA-based therapeutics as potential antifungal treatments, and the second, the role of extracellular vesicles in fungal pathogenesis. And so I think in a medical mycology community, I don't really need to introduce the problem of fungal infections. But of course, fungi infect over 1 billion people per year. The majority of these infections occur as infections of the skin and the mucosa. However, there's also a large number of life-threatening invasive fungal infections that occur as well. And these kill about as many people as tuberculosis per year, or similar to the number of people that died from COVID-19 in 2020. And so of course, this is a major problem, and we really need to deal with this in some way soon. And so the organism that we've been studying for the past few years, and that will be one of the main focuses of my own lab, is Aspergillus fumigatus. This is a spore-forming filamentous fungus. It's found in the environment all around us. And more than likely during this talk, you'll breathe in a few spores. These asexual spores turned canidia, which are released from canidiophores. You can see sort of a depiction of this structure here. These spores are about 2 to 3 micrometers in diameter. And although they have a very complicated cell wall, this isn't really the point of the talk today. All I want to show you is one particular layer. And this is a layer that's due to DHN melanin. This is a pigment in the cell wall of Aspergillus fumigatus canidia, and it gives the canidia their green color. And so we can disrupt genes important for the biosynthesis of this particular layer, for example, by deleting the gene PKSP, and this leads to canidia that are white. And so this is something that will become important a little bit later. And so when these canidia become airborne and float around in the air, they eventually will land somewhere, and if they're able to grow in this new environment that they find themselves in, they will begin to swell, form germlings, and ultimately hyphae that restart this process. If a canidia lands in the lung of an immunocompromised individual, though, we can end up with an invasive infection, for example. And of course, these are quite dangerous. And I think there's a couple of major problems associated with these infections and fungal infections in general. And the two major problems are both diagnosis and treatment. So first off, these infections are often difficult to diagnose because they appear quite similar to bacterial or viral infections that are very common in clinics, obviously. But they also, once we've diagnosed them, we have limited options for treatment. We do have some good drugs, I will admit. The azoles work quite well in treating Aspergillus fumigatus infections, but most of you are quite aware that resistance is increasing, and this is a major problem that we have to deal with moving forward. And so in my lab, we're interested in doing basic research into 3 different directions that will hopefully allow us in the end to make some advances where we can try to use RNA-based therapeutics. And so we're interested in combining the study of fungal infection, RNA biology, and extracellular vesicle biology to try to get to this end. And of course, developing an RNA-based therapeutic is a long-term goal. And so we're interested in just trying to see what we can learn along the way and trying to get to this path. And so today, what I want to do is introduce you to RNA-based therapeutics and talk about how we are thinking about these problems moving forward. And so RNA-based therapeutics are rapidly emerging. And I would say if I gave this talk a year ago, the majority of the people in the audience had probably never heard of RNA-based therapeutics as a strategy. But now you're probably all aware of messenger RNA vaccines that might be being developed by Moderna or BioNTech, because we're hoping that they're going to change the way we've been living our lives for the past year. Of course, this isn't the only type of RNA-based therapeutic that's out there. People have been trying to manipulate a lot of different pathways using the CRISPR-Cas system or RNA interference pathways to try to develop new ways to target genetic disorders or cancer or even viral infection, for example. Of course, there are also many advantages to using RNA-based therapeutics over other forms of therapeutics. For one, they're highly specific. And so, of course, all of you working on eukaryotic pathogens know that if we can use drugs that are very specific and sequence specific, this might open up new targets where normally we've sort of struggled, since eukaryotic pathogens have genomes that are often quite similar to our own. Of course, RNA therapeutics are also active on, in some cases, undruggable targets, and they can be very easy and rapid to design. The Moderna vaccine, for example, the story goes that it was designed in about 2 days' time. So this is quite fast once the tools are in place. They're increasingly cost-effective, and they can be pretty easy to combine in drug cocktails as well. And so hopefully these are all things that could benefit this sort of project in the future. So what I want to do today is talk just a little bit about how people have been thinking about using RNA interference as a pathway that could be exploited for the development of therapies against fungal infections. And so to do this, I have to introduce you to the RNA interference machinery. And this is about as simple a diagram as I could create to try to get this concept across. And so basically, this starts with a double-stranded RNA. Double-stranded RNAs could be produced by a virus. We could make it in the lab and deliver it exogenously to an organism. But when double-stranded RNA comes in contact with a dicer protein or a dicer-like protein in many fungi, the RNA is cleaved into small pieces, which are bound by an argonaute and complex with many other proteins, depending on particular situation. And this particular RNA can then target an mRNA to silence it, usually by cleavage of the RNA, but sometimes by blocking other binding sites. This system can be amplified by RNA-dependent RNA polymerases. This occurs in some organisms, but not all organisms. In a related organism, Aspergillus nidulans, this does not seem to be the case. Whereas other fungi like Neurospora, for example, seem to use RDRPs. And so in Aspergillus fumigatus, we know from work from Isabella Moina and Jean-Paul Leger's lab from about 15 years ago now, that there is a functional RNAi machinery. And you can see that here in this simple example that comes from this paper. And so on the left, you're seeing a colony of Aspergillus fumigatus growing on a plate. And this is green because of this melanin layer that's present in the conidia. Now, if you produce a hairpin RNA, a little double-stranded RNA inside of the fungus that has homology to this PKSP gene, so now we've replaced the target here, you can silence the PKSP gene when this is expressed, and now you have white conidia. And we'll come back to this phenotype in a minute. But we were interested in this idea and trying to understand if we could figure out which RNAi machinery might be important for the silencing reaction. And so we know that in Aspergillus fumigatus, there's 2 dicer proteins, 2 argonaute proteins, and 2 RNA-dependent RNA polymerases. And so this was our question: which Aspergillus fumigatus RNAi genes are important for silencing, and what can we learn about them? And so Raman Kalani, a PhD student in the lab, decided to try to explain this. And so what he did was he made a construct that's very similar to the one from this paper from Jean-Paul Leger. It has a construct that produces a hairpin RNA or an inverted repeat transgene. And so you have a piece of sense RNA specific to PKSP, a linker sequence, and then the antisense sequence of PKSP. And you put a on promoter in front of this, so it was inducible with something like doxycycline and a terminator here. And then he put this on the chromosome of a wild type strain. And so if you don't have the inducer, you can see that the colony is green. And now if you induce with doxycycline, you can see that the colony turns white because you're silencing the PKSP gene. Of course, in a strain that lacks the inverted repeat transgene, you can see that this induction by doxycycline does nothing. So what Raman wanted to do was make knockouts of all the different RNAi machinery, and so this is what he did. And then he put his inverted repeat transgene construct in each of these strains to try to see the importance of each of these genes in silencing. And so what he found with the DCL-A protein, or the dicer-like A protein, was that when you induce with doxycycline, you still get silencing, and this suggests that this particular protein is not important for silencing. When he did the same experiment with the DCL-B protein, he could now see that there's no more silencing. This suggests that DCL-B is required for this silencing reaction. And you could see that here. And when he uses a double knockout of DCL-A and B, again, we see that the colony remains green. And of course, this is likely because of the activity of DCL-B being deleted. So Raman went on and he put these knockouts into all of our different knockouts, or this inverted repeat transgene construct into all the different knockout strains he developed. And he found that DCL-B and Argonaute-A, also known as PPD-A, seem to be required for this type of inverted repeat transgene silencing. He also was able to show that these RDRPs, which seem to be dispensable for silencing in Aspergillus nidulans, are also dispensable for silencing in Aspergillus fumigatus. So now we have a little bit of a better idea how this works. But so the point of today's talk was to try to introduce the concept of developing an RNA-based therapeutic. And so far we've expressed an RNA inside of the fungus, and it seems to have a phenotype. But the next thing that you might want to do is try to deliver an RNA from outside of the fungus into the fungus. And so it's been known for quite a while, and from work from the labs of Hubertus Haas and Alexander Huttenhofer, that this is something that you can do with Aspergillus fumigatus. For example, Aspergillus fumigatus, we've known for a while, will trap negatively charged nucleic acids in the cell wall. And so here I'm showing you a variation of the experiment that they did before, but this was done by a PhD student in Axel Brachhage's lab, Carissa Visser. And you can see the cell wall of the fungus stained in blue with calcofluor white. And it's a little bit hard to see maybe in this picture, but in green is a double-stranded RNA that has been in vitro transcribed using fluorescein labeled UTP. And so a fraction of the uridine residues are labeled with fluorescein, and you can see that in green. And unfortunately, what you can see is that the RNA ends up trapped in the cell wall of the fungus here. And so despite this, you can actually get some level of silencing if you have a particular target gene. And so we do get some knockdown, but it's not so much. And you can try to enhance this response by adding some liposomes, something like lipofectamine, to encase the double-stranded RNA and try to deliver it more effectively to the fungus. But this doesn't really change the situation. You still have a lot of double-stranded RNA trapped in the cell wall. And so, of course, this is interesting, but maybe we need to be better at designing RNAs to deliver to the fungus, or we need better tricks to get the RNA inside. But in some cases, incomplete knockdown might actually be enough. And so people have tried to see if this is the case. Can we use the knockdown that we have as a potential tool? And I'll just show you one quick example of this. And this is looking at CYP51A, which I think many of you are aware is a major source of resistance for Aspergillus fumigatus. And it's a target of the azole drugs. And so in a wild type Aspergillus fumigatus strain, if you treat with an azole like itraconazole, there's a low minimum inhibitory concentration, meaning you can kill the fungus with a low dose of this particular drug. It produces some CYP51A, and this is the target. Now, in a mutant strain, we won't cover how this strain is mutant, but typically it's an overexpression of CYP51A, which leads to higher tolerance for this particular drug. And you can see in this experiment by Bita Musavi and al, that they see higher levels of tolerance, up to 16 micrograms per mil. Now, if they add an siRNA against CYP51A, they can knock down the levels of CYP51A in this strain and then restore the sensitivity to the azoles. And of course, this is really interesting because it means that if you have a patient that has developed a resistance, you might be able to just quickly design an RNA treatment that could knock down some of the resistance mechanism, if it's a mechanism that's similar to this sort of pathway. Of course, there's many things that we could do to try to improve this. We could design better RNAs to bind more robustly. But today, what I'd like to talk about in the last few minutes is how we might improve this by improving RNA delivery. And so one thing that we know from our friends in the plant fungal pathogen community is that extracellular vesicles are often a way that RNA seems to be delivered from a plant host to a fungal pathogen. And this is beautiful work from Haling Jin's lab at UC Riverside. And what they were able to show is that the plant is able to send RNA, particularly small RNAs, that target fungal virulence genes to plant pathogenic fungi. The fungus will also actually send RNAs back the other direction. But the really interesting result is that if you spray double-stranded RNA on plants, this double-stranded RNA will be packaged into EVs or possibly directly transferred to the fungus. And this can target virulence genes in the fungus and really improve the outcome of the infection for the plant. So this is really interesting. And so for the last few years, we've been focused on extracellular vesicles in my time in the lab of Axel Brachhager, and it's something that we're still interested for my lab as well. And so I wanted to at least introduce extracellular vesicles here. This is a very heterogeneous population of small nanoparticles that are produced by a variety of different mechanisms. So some are produced by multivesicular bodies. Typically, we call these exosomes. And you can see that the size range goes from about 30 nanometers up to about a few micrometers in diameter for these various different particles. Others are produced by budding plasma membrane or blood-being apoptotic cells, but really this is a heterogeneous group that's quite difficult to study. And so a number of years ago, a former PhD student in Axel Brachhager's lab, Jordana Shapova, was really interested to understand how polymorphonuclear leukocytes that produce extracellular vesicles might be contributing to the outcome of Aspergillus fumigatus infection. And so since this time, we've tried to expand the cell types that we are studying and develop additional methods to try to understand how these extracellular vesicles might be contributing to infection. And so we typically isolate extracellular vesicles using a combination of centrifugation and filtration, as well as occasionally we use size exclusion chromatography to really create clean, pure populations of these EVs. And then we can measure them using a lot of different methods, imaging flow cytometry or Western blots to look for different markers, cryo-TEM, for example. Today, I'll just show you one example of how we can use nanoparticle tracking analysis to measure the size and concentration of these particles. And so you can see that here in some data from Flora Riviecchio. She's looking at A549 epithelial cells. And so now you're looking at the concentration on the y-axis and time on the x-axis. And you can see that over time, epithelial cells produce EVs spontaneously, that's the black bars. And when we look at fungal-induced EVs, you can see that it increases over time, but it's not a major difference between these two populations. If we look at the size distribution of these EVs, again, concentration on the y-axis, you can see that the size distribution of the two EV populations, the spontaneously released and the fungal-induced EVs, are quite similar. And this is quite similar for neutrophils as well when we look at primary neutrophils. And so here, Anne-Katrine Zimmermann was doing a similar experiment using the NTA. And again, you can see that over time, the populations are pretty similar in concentration, and also they're pretty similar in their size distribution. Now, they are a little bit bigger, and this is known for neutrophil-derived EVs. So Jordana Shapova was really interested in trying to characterize the different cargo that were present within these extracellular vesicles, and she was able to show by flow cytometry that there were different subsets of EVs produced. And by LC-MS-based proteomics, in collaboration with Thomas Kruger, she was able to show that fungal-induced EVs actually have quite a different content. So a number of proteins are present only in fungal-induced EVs and not in spontaneously released EVs. This is in stark contrast to epithelial cells. And in this case, you can see that the majority of the proteins that you find in these extracellular vesicles are found in both spontaneously released EVs and fungal-induced EVs. So of course, Jordana was very interested in trying to follow this up and see what this might mean for their function. And so what she found, I'll just show you two quick results. The first is she was able to show that the extracellular vesicles, when stained with pyridium iodide, would localize to the cell wall and then in some cases enter into the cytoplasm of fungal hyphae. And you can see that stained here. In green is the cytoplasm and in blue is the cell wall stained in calcofluor white. This suggests that the EVs at least associate and in some cases might even enter inside of the fungus. And so this would suggest that perhaps these EVs were antifungal. And this was something that is very well outlined in Jordana's paper, and I suggest you go there to get more details, but I can give you one example of how we tried to test this. And this was using a mitochondrial GFP reporter strain that was produced by the lab of Johannes Wagner. And in this particular strain, you have a mitochondrial network that's labeled with GFP. And in live hyphae, you can have a very nice filamentous network of this of these mitochondria labeled. And when you challenge them with H2O2 or something that will kill the hyphae, now you have fragmentation of the mitochondrial network. And so this makes it very easy to see. And in fact, when we look at this biconfocal laser scanning microscopy, you can see that in untreated hyphae, now the mitochondrial network is labeled green, and we have this nice filamentous network. In H2O2 treated hyphae, you can see that the mitochondria have fragmented, suggesting that the fungus is in fact dead. When you add spontaneously produced EVs from neutrophils, these have no effect on this reporter strain. The filamentous network is intact. However, fungal-induced EVs, we can see now kill the fungus. So there's something about this fungal challenge of neutrophils that results in EVs that are produced that are antifungal, consistent with the proteomics and the other results that I've shown you. The interesting part is that if you use EVs that are produced in response to a PKSP mutant strain, this is the fungus lacking this melanin layer, so these white spores, now these particular EVs produced are no longer antifungal, and you can see this filamentous network present in the hyphae. So this suggests that there is some specificity, and maybe melanin is one of the signals that leads to this antifungal activity. And so there's a number of questions that this leaves us with. We're really interested in trying to understand what is the antifungal cargo of these extracellular vesicles. Of course, my lab is really interested in understanding this from the perspective of RNA. We're interested if there are extracellular RNAs in general, either within or associated with EVs, or possibly just bound in RNPs floating in the media, for example. We also are interested to see if we could develop these EVs as useful diagnostics for fungal infection, particularly looking for maybe RNA targets. And the whole reason we started talking about extracellular vesicles is that we're interested to see if we could use these EVs or some knowledge that we gained from their study to be able to better deliver RNA into the fungus. And this would be a sort of a long-term goal. And so really the question at the end is there a future for antifungal RNA-based therapeutics? And I think, of course, the jury is still out, but I think there's a lot of potential here. I think we need to do more to understand the targets that we're using, maybe develop new targets and new therapeutic options. Of course, we're going to need to be able to deliver the RNA directly into pathogens and not just have it cleared in the kidneys or liver, for example. And in the long term, of course, like everyone, we'll have to minimize off-target effects and host nucleic acid sensor activation. So although there's challenges, I think there's paths forward, and I think our lab is well positioned to try to make some progress in this field. And so I hope in the end that I've told you that RNA-based therapeutics could offer a path to new antifungal treatments, and it's something that we need to at least consider, and that extracellular vesicles seem to be important players in fungal pathogenesis. And with that, I will thank the members of my lab. Although small, I think we have a great team that we're building quite quickly. I'd also like to thank Axel Brachhager and his continued support, and especially the members of the current EV crew, Ani, Flora, and Carissa, for all their great work. Our collaborators, Johannes Wagner at Trinity College Dublin, as well as the Federal Ministry of Education and Research, who's funding our work for the next 5 years, which is fantastic. So if there are questions, of course, I'll answer some later. And if you have others, contact me on Twitter or via email. So thanks for listening.
All right, thanks, Matt, for a great talk. If you have questions, please enter them into the chat. We'll take them all at the end. And now I'm going to pass it to another member of the organizing committee, Marcia Rodriguez, who's going to introduce Carolina.
Everybody, so it's my pleasure to introduce today's next speaker, Carolina Coelho. It is also great to see so many friends participating today. So Carolina was born in Portugal, where she completed a pharmaceutical sciences degree in 2007. And in 2008, she joined a PhD program in Portugal, where she was supervised by Teresa Gonçalves in Portugal and Arturo Casadeval in the US. At that time, Arturo was in New York at the Albert Einstein College of Medicine. After completing her PhD, Carolina moved to Baltimore when Arturo moved to the Johns Hopkins University. And since then, Carolina has been always working with models of related to the interaction of Cryptococcus with different hosts. Carolina is a great immunologist. She's also committed to increasing the awareness of fungal diseases, which is highly appreciated. And today, Carolina is going to talk about the interaction of Cryptococcus with mammalian hosts, which is, of course, the best model ever. So, Carolina, thanks a lot for participating, and the audience is all yours.
Thank you so much for the invitation. Thank you so much for everyone who's here. It's great to see so many new people to the community and also revisit some friends, even if remotely. Thank you so much, Marcio. I just started my lab like Matthew. I'm quite new. I work right now at the University of Exeter in the Center for Medical Mycology. And obviously my focus is Cryptococcus neoformans and how it interacts with the host. So the first thing I will say is that Cryptococcus is the top fungal killer, is the fungus that kills the most humans. This amounts to about 180,000 deaths each year with about 70% mortality. And in this graph here, what we're showing is that this is really due to the burden of HIV/AIDS, which unfortunately is higher in developing countries due to lack of antiretroviral and anti-AIDS drugs. But the important thing to highlight is that this mortality rate is still very high, even with access to the best medical care. If you are immunocompromised and you have access to the best medical care, you are still probably having a high mortality rate and very serious, long-lasting damage from a Cryptococcus infection. So a lot of people survive cryptococcal infection but are left blind or death. So it's really important that we continue to investigate this pathogen so we can really improve the outcomes for the patients. So let me introduce you here, meningitis caused by Cryptococcus. As you can see here, the white masses are all Cryptococcus growing in a brain. This is a brain scan of a very sick patient. As you can see, instead of being a sort of symmetrical structure that we would expect in a human brain, what we see is a black mass that has shifted the brain. So this is someone with a really serious brain infection, Cryptococcus. And you can see here is pathogen Cryptococcus. It's a very photogenic yeast, very pretty, very circular with little buds coming out, the daughter cells. And this halo is obviously the capsule, the sugar capsule that looks like fibers, long polysaccharide chains that are really a mediator of the interaction with the host. So this is Cryptococcus. So the other thing I would like to highlight is that classically, we've been talking about Cryptococcus neoformans as the agent, the etiological agent of cryptococcosis. But in recent years, that view has changed. And for example, I will tend to talk about Cryptococcus neoformans, but the truth is we now know that a lot of cryptococcosis is caused by other fungi of the same genus. So for example, data in humans, in HIV patients in 2 different countries have shown that up to 30% of the cases can be caused by Cryptococcus gattii. And the same is true in Australia, in cats in Australia. 30% of the infections can be caused by gattii. So really the field has now shifted to talking about Cryptococcus pathogenic species, Cryptococcus species complex as the causative of cryptococcosis. And we are still investigating how these similar species, how these very closely related fungi cause disease and how they relate to each other. But I would just really like to highlight that these seem to be very closely related. And so far, they cause the same disease, at least in a very broad sense of the word. So Cryptococcus, all of the species, they exist in the environment. They're associated with birds, they're associated with trees, and from their environmental niches, they become airborne, either the spores or dried yeast particles. We inhale them, the same as Matthew was saying. The airways are kind of like our exposure route to a lot of these fungi. And from there, they deposit in the lungs. And if you're healthy, an immunocompetent person, a healthy immune system will fight this infection, kill the pathogen, or at the very least control its growth. Causes a late infection, no problem at all. However, if you become sick, immunosuppressed, such as unfortunately having AIDS, or if you undergo a transplant, the immunosuppressive effects will allow the crypto to start growing, the fungus to start to grow, and you're going to have either a pneumonia or a meningitis. And as I said, it's incredibly serious and incredibly hard to treat. The disease is always an interaction between the host and the pathogen, and they're fighting. Obviously, they're fighting for survival. So the fungal pathogen will try to grow, will try to invade and disseminate, and the mammalian host will try to kill the host. And they both use tools, fungal fitness and virulence factors, while the host is using its epithelial barriers, other types of barriers, immune barriers, particular macrophages and T cells are really the cells that interrupt the crypto and that try to control it. But this disease is really a tug of war. So obviously it makes sense that if I want to understand this disease, I have to study the host and the pathogen, how they interact. So in my lab, what I try to do is to study the fungal invading factors and also the immune system, and in particular the macrophages, and in the recent years, the immunometabolism. And this is something that I'm not going to speak about a lot today. Hopefully I'll give you guys another talk in the future, and I'll talk about immunometabolism. Right now, I will focus today on fungal invasion factors. So a lot of the tools that we use are mouse models in order to mimic a disease, an interaction. And we would take Cryptococcus fungus to Cryptococcus yeast, and we would inject it, infect mice. We can either do an intravenous infection where we inject directly into the bloodstream. This is going to cause a very quickly disseminated infection because in the bloodstream it will quickly disseminate. Or we can do an intranasal infection where we deposit the yeast inside of the nostrils. We're allowing it to interact with the airways, get deposited into the lungs, because we think that is going to mimic a natural infection a little bit more, going to cause an upper airway infection. And after the mouse is infected, we are either going to monitor mouse survival, how the mouse gets sick, how much weight they're losing, or we can also measure how many yeasts we have in each of the tissues. So we would extract the tissues. We're going to measure usually burden in the lungs and in the brains because those are the target organs. And we use that as a surrogate of how the infection is progressing. And lastly, obviously, we're going to use gene deletion strains, usually for the yeast, but it could also be for the mouse. Models where we're going to try to isolate a factor, we'll delete the gene. We're going to reconstitute the gene, put it back on. We're going to try to isolate the effects this gene has. So one of the well-known factors of Cryptococcus neoformans was urease. Urease is an enzyme expressed by Cryptococcus neoformans very high amounts during infection. This is an enzyme that degrades urea into ammonia and carbon dioxide. So because it plays such an important role in infection, when you delete urease, what you see here, the open circles, is that when you delete urease, the fungus cannot kill its mammalian host. So in a survival curve, the mice would slowly die of Cryptococcus from the wild type strain, but not if you delete urease. And the same is true for an intranasal infection, where if you delete urease, the mice survive longer. They have a better chance of fighting this infection. For many years, this was known, but the mechanism was still unknown. We didn't know the role that urease was performing, how urease was contributing to killing the mouse. Curiously, what was known from very early days is that urease is required for the fungus to survive in the tissue. But and I can show you here that data where both in the lung and in the brain, if you infect with urease deleted strain, what you see is that the urease deleted strain has less CFUs, less fungi compared to the wild type strain and to the reconstituted strains. So the hypothesis one was that urease helps Cryptococcus to survive in the tissue. But we had no known mechanism. We didn't know what urease was doing. And at the time, in Arturo's laboratory, we were thinking about this, and we said, you know what? We have an alternative hypothesis. It could be that urease helps Cryptococcus survive the immune system, and the most important cell of the immune system to kill Cryptococcus are macrophages. So how about we test the survival of urease deletion mutants in macrophages, and our prediction would be that urease deletion causes decreased survival inside macrophages. So I did this work with Sarah Fu, Manshun Fu, who was a wonderful worker and a wonderful colleague in Arturo's lab, and she's the main author in this paper that I highly recommend that you will go read, and I'll just show you the highlights of the story. So what we did at this point is that we took Cryptococcus, again, wild type deletion strain for urease, and the reconstituted strain, and we exposed it to murine macrophages. Cryptococcus is ingesting the phagosome, exposed to an acidic environment, among many other things. And then we investigated how the survival of the urease negative strain in this environment. We were very surprised by the result, because actually what we saw was that our hypothesis was that instead urease deletion caused an increase in growth inside macrophages. It had a higher number of CFUs compared to the wild type. So our hypothesis was rejected. And we really thought about this for a little bit, and we couldn't really figure it out because we knew there's less, if you delete urease, there's less survival in the tissues. We figured at some point, we remembered that the pH inside the macrophage phagosome is acidic, 4.5 around 6, and the pH in tissue is 7.4. So we had an alternative hypothesis, formulated an alternative hypothesis where urease helps Cryptococcus survive depending on the pH at different pHs. So we tested that, and just to show you this data, this is growth at very acidic pH of a wild type strain. As you can see, as you slowly increase the pH, you're going away from the optimal pH of Cryptococcus, which is fairly acidic, and it's growing slightly slower as you increase the pH to 5.6. However, the urease negative strain grows less when the pH is becoming less acidic, when it's becoming more neutral. And this really showed us that our second hypothesis was correct, is that urease deleted strain seemed to have trouble when the pH was becoming neutral. So then we decided to test what would happen at physiological pH, because mammals are usually pH 7.4. Bloodstream or tissue are usually at pH of 7.4. So when we expose the urease deletion strain here in yellow to a pH of 7.4, it grew much less than the wild type strain here in blue. But this really shows that the urease is needed for optimal growth at a pH above 5.6, and particularly at the pH found in mammalian tissues, which is really important also because it shows that there's also a contribution of the extracellular stage, that the urease is enzyme action is within the extracellular stage of growth, and urease is required for optimal growth. Apparently not within macrophages. So as I showed you here, this is the fungal invasion factors, and I started to become more and more interested in deciphering these. And another observation that we made immediately after was actually we needed to really understand the kinetics of when Cryptococcus was invading and disseminating, because we went through the literature and we just assumed that there was a latent infection in the lung, and then crypto would overgrow and only then it would disseminate into the brain. But we actually had no hard data for that. So we went and started to look for that, partly as a way of setting up and starting to really develop these models of invasion in the mice. So in this particular experiment, we did compare 3 different types of infection. We took Cryptococcus and we did an infection into the bloodstream, the intravenous directly into the bloodstream, compared the lung infection intratracheal deposited into the trachea. And then we also did the intranasal where we deposited in the nostrils. For all of them, we compared the number of CFUs, the number of yeast cells into the lung and the tissue as a way of measuring how quickly the crypto disseminates, how much it disseminates. And to our surprise, as predicted, we saw that the intravenous infection was faster than the other infections. So at 3 days, you have higher number of CFUs in the brain if you infect with the intravenous route versus the other routes. But the truth is, there was still a lot of Cryptococcus in the brain at this point of infection, 3 days post-infection. And the idea beforehand was that you had to have full lung infection before you had a brain infection. So we just looked at each other and we decided, you know what, let's go and measure a really short time course, because perhaps crypto is going into disseminating early, and it's disseminating into the brain much earlier than we thought previously. So we did that experiment, we repeated that experiment, but this time we did an intranasal infection, and we measured tissue burden at 3 hours and at 24 hours. And I can see even in an intranasal infection of 3 hours, you already have yeast cells inside the brain. So that really shows how remarkable this invasive capacity is. 3 hours after you have exposure to Cryptococcus in the nose of an animal, you can already detect Cryptococcus inside the brain of the animal. So really remarkable invasion strategies that we are going to try to investigate further and really elucidate in the laboratory. And the last observation that we did for this paper is that we observed the nostrils of the animals, and this is the sagittal cut. So this is nose to bottom of the animal. And you can see here the nose cavity. This is the brain, and here below is the mouth of the animal. This would be the roof of the mouth. And here is where you smell. This is the olfactory epithelium. And when we zoom into this area, what we already see is a ton of crypto. Crypto is these round circles here, a lot of crypto in the nostrils of the animal. So really, all the upper respiratory tract is infected with Cryptococcus. So we're very happy about this. We published this data, and actually we got a wonderful email from Richard Malik, who's a researcher and veterinarian in Australia, who wrote us this email. Thank you so much, Richard. And who told us that this data in mice actually really replicated his clinical data, where he knows that cats get nose infections with Cryptococcus. Koalas do too. So really an entire upper respiratory tract, also lower respiratory tract infection caused by Cryptococcus, underscoring then very rapid brain dissemination using mechanisms that we don't fully understand yet. So I'm getting to the last slide of my talk. Just wanted to give you the summary, the take-home message. I just showed you that fungal urease is required for growth at neutral pH. This is why urease is required for growth and full virulence in Cryptococcus. We don't know the molecular mechanisms yet, but we already know the cellular mechanisms. I've also shown you that brain invasion of Cryptococcus is incredibly fast, and that Cryptococcus also infects the upper respiratory tract, the nostrils, and the ears of animals. And that mostly a lot more needs to be understood about invasion mechanisms of Cryptococcus. And obviously that is something that I would like to understand more in the future. And as I said, just to highlight, I'm also very interested in how the immune system interacts with Cryptococcus, particularly macrophages and their immunometabolism, but that is a topic for another talk. And then lastly, my acknowledgements. I would like to thank all of my collaborators, my friend and my brilliant colleague, Manshun Fu, whose work I showcase here, Arturo Casadevall, who was my postdoc mentor, and now the Center for Medical Mycology, all of my wonderful colleagues there who are really allowing me to grow my lab and research ideas. My students who've done some of this work, Antonio, Leo, Daniel and Dimitro, Lucy also, who I have as a collaborative student, funding. And then I would also like to highlight that the Center for Medical Mycology is recruiting for PhD students right now. And you would have an opportunity to do work with me or with anyone else in that Center for Medical Mycology, a really good place to work. And with that, I end my talk. Thank you so much.
Thank you so much, Carolina. That was great. So Jessica, if you want to take questions for Matt first, and then I'll get Carol's questions.
Sounds good. So if everyone would please enter questions for Carolina and Matt in the chat. I'll start with a couple of questions for Matt. And the first one is from Brenda Bass, which is, do you know what the cargo is in the fungal infected EVs?
Yeah, so we've been doing a lot of proteomics over the last few years to try to understand from a protein perspective what's in the EVs. And it really depends on the cell type. For the neutrophil derived EVs, it's sort of a list of antibacterial and antifungal components that you might expect. So neutrophil elastase and I think histones and myeloperoxidase, lots of things that we might expect would be produced in a neutrophil. For the RNA cargo, we don't really know. This is one of the first experiments that we're doing now that my new postdoc is setting up. And we're really interested to see. We have some guesses what might be there, but we don't know exactly yet.
Thank you.
And a question from Marcio, which is, do you have any idea about the fate of mammalian vesicles in fungi? Do they interact with the cell wall? Do they have an intercellular target? Anything like that?
I wish we knew. So in Jordana's paper, she did a lot of work trying to localize where the EVs, or where the majority of the EVs end up. It seems that most end up in the cell wall. There's a smaller fraction that make it sort of to the membrane, and then an even smaller fraction that you could see maybe completely inside of the fungus. We're really interested to try to figure out what the targets are, but it's not such an easy problem, unfortunately. But I think we're getting a little bit better with some of the tools that we're now making to try to answer this question.
All right, do we want to take a couple of questions for Carolina and then we'll go back and forth?
Yeah, sure. Carol, the first question that we have here is from Carolina Paul. She's asking if you see any behavioral effects, especially during the early stages, first few hours in infected mice, which is actually a great question.
So I can tell you that when I'm comparing with PBS, when you have to do with a PBS control with a vehicle, and the crypto, the very first reaction that they have while they're waking up from anesthesia, it seems to be different for Cryptococcus. It's not the same as for PBS, right? That said, after a couple of hours, no difference. If I was blind, I don't think I would see a difference between the infected animals and the PBS control, the mock-infected animals who have gone anesthesia and all of the appropriate controls. So absolutely not. And the amount of fungi that they have in the lungs is already astonishing. We know from other studies, from previous studies, that at 4 hours, there's already a ton of neutrophils that are responding to the inflammatory stimuli. So after 4 hours, you already have neutrophils and inflammatory monocytes into the lungs, but the mice are behaving completely normal. Perhaps if we're measuring, obviously their cytokines are already changing, so perhaps we would need a really detailed study, which is difficult with a small model. But yes, interesting question. Nothing gross, I would say.
Thank you, great. We have a question from Zia Guza. She's asking, or maybe he's asking, I'm sorry. In C. diniformans, a less virulent species, we also found dissemination to the brain in about 10 days using the retroorbital model of infection. What are your thoughts on using this model for assessing cryptococcal infection when the natural route of infection is intranasal?
I think every model serves its purpose. And for example, I've used the IV as a way of quickly inducing brain infection because it does go faster. I think if you have a model that needs the IV infection because you don't care about the interactions in the lung or you want them all to happen at the same time, I don't think that we know enough to completely discard the IV infection model. And I know, for example, a lot of the interaction within the endothelium, they use the IV model because they want to have enough to image. I can't see why not. So every model serves a purpose, and intranasal is not perfect either, right? Because it's not a dried particle. When instilling it suspended in liquid, it's not a dry particle. I know there's work in spores, which are perhaps the more natural infectious particle. So I don't think we have any data that says that IV is bad. So go ahead and use it if that's what you think is good. Perhaps worry about doing one experiment in intranasal just to validate the main finding.
Thank you, Jessica. Great.
So Matt, a question from Gustavo Goldman. Have you checked altered phenotypes for the RNA pathway mutants? For instance, the susceptibility status for azoles or caspofungins?
Yeah, so we've done a lot of work trying to find phenotypes with these mutants, and to be honest, it's mostly underwhelming. We do have a couple of leads, but in general, we've tried to do some of the azole susceptibility sort of assays with the dicer knockouts. This was quite a while ago. We didn't really see any obvious phenotypes, but we've bought some new drugs that we'll try again soon now that we have this sort of full panel of knockouts. This is, of course, a good idea. Yeah, we've been trying lots of different things, but we haven't really found any great phenotypes at this point.
Okay, in that case, I'll butt in with my question since that's a good segue, which is, does treatment with a cell wall damaging agent like an echinocandin release any of the possibly charged nucleic acids that are sequestered in the cell wall?
This is a good question. I don't know the answer. In the literature, they've shown that these, at least in one of these papers, one of the RNAs they were looking at was a long double-stranded RNA, and it seems that it gets cleaved by something like a dicer protein and internalized. And so it could be that by using an echinocandin or something like this, that you might cause all kinds of changes by disrupting the localization of all kinds of things. But it's a good idea. We could try to do something like this.
All right, and a question from Jorge Meek, which is, what would be the approach to deliver the RNAi containing extracellular vesicles to the infected lung? Synthesis in the lab and direct intranasal delivery or nebulization? Maybe trying to express the RNAis in immune cells so they produce extracellular vesicles, or other such ideas?
Yeah, I mean, I think both of those ideas are interesting. Of course, anytime you are nebulizing something into a lung, I think you have to be careful that you don't have sort of a crazy immune response in response. And we know that pieces of RNA, especially small RNAs, if they get out of hand, can activate these nucleic acid sensors, which could lead to some sort of terrible response. And so I think that approach, it could work if we have it encapsulated in the right thing. But in general, I don't know the answer. I think we'll have to see where the field goes, what we can do.
Great. Shall we take a few more questions for Carolina now?
Sure. Carol, a question from Vikas Yadav. Have you tried to test if the early infection rate varies between different species, mainly between Neoformans and Gattii? And this is something that I would like to ask too. I would just ask about different isolates instead of different species too. Is there, the variability, is it too high?
So in the paper, we actually tested H99O, which is supposed to be very similar to the original isolate of H99, which, as you know, is a Neoformans pathogenic. We also tested R265, which is Gattii, and the outbreak strain, the British Columbia outbreak strain. They all got into the brain. If you have differences, it's related to the virulence of that strain. In particular, it's not directly related to if it's Neoformans or Gattii. So R265, yes, it goes into the brain quickly. So I think it's strain related, and they all migrate and disseminate quickly.
All right. Felipe Santiago Tirado, nice talk, Carolina, very clear and easy to follow. He's curious about the intratracheal and the intranasal models. Both had similar brain burdens at 3 days. Yet you see brain burden in 3 hours in the intranasal. Did you try the intratracheal in 3 hours? I'm very curious why the intranasal gets so fast in the brain, yet it levels off as the same, and it is the same with the intratracheal after 3 days. Did you get it? Because I made some confusion. Was it clear, Carolina?
No, it's fine. So we wonder about. Is it clear? I lost you, Marcio. So I think I can answer the question. In the first set of data, 3 days, what we saw is no difference between the intratracheal and the intranasal. So for the other experiments, we only use intranasal. Part of the reason is less anesthesia. So if we're measuring short time points, you don't have to worry so much about effects of the full anesthesia. And also no damage to the mucosa because it's a less invasive procedure. So I think it's a little bit better for 3 hours just because the mice are recovering fast and kind of normal faster. And we saw no differences. The intratracheal, if I think technically, it's a little bit more precise in the way you deliver the infection. It's a little bit easier to deliver the right amount of fungi. but we didn't see a difference between them, which is why we ended up not repeating for the intertracheal. We had no data that suggests that intertracheal versus intranasal was going to be very different.
So Jessica, if you don't mind, because the next question for Carolina is really related to this one.
Absolutely, go for it.
Yes, I think it's really related, just to keep in the same topic. Do you think the intensity of the disease would be greater or more comparable if you did an intratracheal infection? Would it affect the CFU in the lungs, brain in some way that would point to a distinct effect in mice?
I don't think so. And then the thing is, again, as I was doing this work, the truth is mice cough and mice sneeze, right? So once they have something that's irritating the lungs, it's quickly going to be in the upper respiratory tract, even if you have deposited it into the trachea. So it makes me think that the intranasal intertracheal starts to become technical, a little bit more anesthesia, a little bit more invasive for the mouse. And of course, perhaps a little bit more difference or a little bit more difficulty in making it precise. But if they cough and sneeze, the upper respiratory tract is going to be contaminated very quickly, right? And the vets are absolutely, of course, that's going to happen. And then, so I don't think it's different. I have no data to tell me that it's different.
All right, I'll take a few questions from Matt. Before I do that though, reminder that the registration for the virtual Candida and Candidiasis meeting is coming up, and I've put the link into the chat function. So it should be a great meeting, and please look it up if you're interested. So a question from Kathleen Mills, Matt, is the release of EVs from neutrophils regulated by canonical antifungal pathways such as C-type lectin receptors?
Yeah, this is something that Axel Brockhager's group is really interested in trying to explore more. We, to the best of my knowledge, haven't tried anything yet with this, but it's on our to-do list, or on the to-do list. Of course, if a receptor like MELEC or something like this was involved, this would be really, really interesting. And so, yeah, I think it will be done eventually.
All right.
To do.
And Chloe Peltier asks, is EV release seen across different fungal pathogens? Do you think there would be common EV contents that might be exploited?
Yeah, I think this is likely the case. There's not a lot known from the host side of things. Of course, there's been a lot of great work looking at fungal produced EVs, and I think there's a related question to this as well. But in terms of host response, not so much has been done. We've been trying to figure out how specific these EVs that are produced against Fumigatus are for other pathogens, and it seems to be very specific is our initial sort of response. And so the data is not quite ready to go yet, but it looks quite good that they seem to be produced in response to a specific pathogen and not, let's say, a bacteria, which is a little bit surprising because the cargo is quite diverse. So this might also offer a reason why an RNA could be a target that's very specific for one type of organism.
All right, and then Alex Poberskin asks a question, but says that as a new person in the fungal field, it might be a little out there. And I just wanted to say that sometimes those are the best types of questions because bringing in a new perspective is always welcome. So thank you for asking the question. And the question is, in the context of issues with RNA delivery, can you perform an assay that uses engineered microviruses to deliver the RNA to avoid trapping the nucleic acid in the cell wall? Right.
I think this is a really good question. I will admit that I don't know so much about mycoviruses, but my understanding is that you often don't get, it doesn't quite work the same as a virus that would infect us that might come from outside. Usually they're transferred laterally. So you don't really have a viral particle necessarily, maybe in some cases. And so I think if we did some more work to try to find viruses like this that are infecting Fumigatus, this could work. But I think of the viruses we know for Fumigatus, we don't have anything like this, but it's definitely an interesting pathway. We've thought about trying to do something like this.
And then a related question, which is, could you use fungal EVs to transfer, to deliver RNAs to fungi?
Yeah, could be possible. Not something we've tried, but definitely a good idea.
All right, shall we take the final questions for Carolina?
I think we are all set.
All right, in that case, I have a question, and that is, do you think that urease might be important at different stages of the infection? Like potentially more important for establishment when the tissue is still at a nice alkaline pH, and maybe less important for dissemination, or?
Absolutely. One of the things I didn't highlight, because I didn't want to start bringing too many concepts, was it seems to provide a role that the extracellular stage of infection is as important as the intracellular stage of infection, right? For sure. That said, in the end, if you allow enough time, it delays the infection, but it doesn't completely prevent urease-deleted strains from killing the mice, right? But that has been happening for all the invasion factors. If you took NPR1, PLB1, they delay the infection, but they very rarely lead to a virulent strain, right? So a lot more to be known. Definitely, I think it does support that the extracellular stage is important, for sure. It seems to be a mixed bag. In vivo tracking the mouse, not as a single time point, but being able to track a mouse across all of the new tools that are developing are going to allow us to answer that.
Great. All right, thanks everyone for coming. We'll have the talks up on YouTube, and if you're interested in presenting, please submit an abstract, and we hope to see everyone next month here, but also at the Mycotalks in a couple of weeks. So great, and thank you again both to Carolina and Matt for two fantastic talks.